Hello, everyone. Welcome to the Winter Quarter CDM Research Colloquium series. For the research seminar today, it is my great pleasure to introduce Dr. Todd Smith, who is the head of business and software development at Digital World Biology in Seattle, Washington. Dr. Smith received his BS from University of Minnesota in genetics and biochemistry, and PhD from the University of Washington in Seattle in medicinal chemistry, followed by completing a postdoctoral fellowship at the University of Washington. Dr. Smith comes to us with a wealth of industrial experience. He founded Geospiza, a company that focuses on developing best practices and bioinformatics software applications for life scientists. Geospiza software systems have established an international reputation for usability and performance since 1997. He was also a senior leader in research and applications at PerkinElmer, which is an American global corporation focused in the business areas of diagnostics, life science research, food, environmental, and industrial testing. Currently, he is the CTO and director of Biotech Careers and Digital World Biology. I know Todd is going to talk more about it, but briefly, Digital World Biology focuses on creating mobile applications and instructional materials that help students investigate molecular biology in the same way that life scientists do. Even elementary school students can use these apps to work with DNA structures and molecules. The overarching goal is to take bioinformatics out of the laboratory and putting it into the classroom. Digital World Biology also offers professional development for college instructors, high school teachers, and also provides consulting services in genomics. I'm glad to have Dr. Smith at our colloquium today to talk about biotech careers in software and computational biology. Welcome, thank you for your time, and please take it away.
All right, well, thanks, Drew. Yeah, we met when you were in New Mexico, and I was doing a different project with a company that just didn't really go anywhere, but it was genomics and pangenomes for a plant that's become very popular in recent times. And I don't know where that was a really great introduction. It must be on the internet somewhere. So with that, I'm going to share.
I wrote it out by myself.
Oh, very good.
No, I grabbed most of it from your LinkedIn and the Digital World Biology stuff. So.
Yeah. No, that was awesome. Now, where's the darn? We just did a test. Maybe, it's crazy. Last time I had Keynote in front. Maybe that helps. There we go. Back to share. There it is. Okay. Find my app. Put it into play mode. Yeah, so when Thura reached out and said, talk about how you use computational biology in your work. I was like, okay. Well, that's kind of a big project because as you mentioned, I've done a lot of different things. And so it's affected my life in different ways. So I thought what I'd do, and it was in the abstract, is kind of talk about how I got into this because I came in from a different perspective, I think, than a lot of people come into computer science. They begin by liking computers. Back when I got started, I didn't find them particularly fun. I'd see people, in the student union debugging code by looking at reams of paper and, and doing punch cards and things like that. And it was like, why do you do that? And so it was really 1984 when the Macintosh came out that I said, okay, now I'm in. So what I'm going to do is kind of take you through a journey of my background, beginning how I got into this at Genetic Systems in the mid 80s when I was a technician. Then I went back to graduate school and got a PhD, did my postdoc, and I often tell people I had the great experience of having people write books, not about me, but the companies and people I worked with. And so I could vicariously enjoy some of my experiences through other authors. And then kind of mid-career, Geospiza, I founded that based on a lot of work I had done in my postdoc, where we got into doing limbs and genetic analysis and these kinds of things. And eventually Geospiza was acquired by PerkinElmer. But, a couple of significant things. First of all, this tidal wave never happened. We always talked about the tidal wave of data, and people still use this as a metaphor, and we're still just getting more data. So that's just not a big problem. And then Finch TV, which I have sort of on the side, this was for Sanger sequencing, but it's that kind of thing where you take a common problem and you make the software useful. Like, you could see a trace in a full screen, you could do drag and drop, you could do very, desktop kinds of things you'd expect to do. And we still have downloads from our site, about 300 downloads a week. It's really quite amazing. It's one of the lasting legacies of Geospiza, actually, because after PerkinElmer acquired everything, they kept the software going as long as possible, but it's sort of all gone now. But and then so that's the first part. And then the second part, I'm going to give you an example of a course I was involved with developing. But I've really been working a lot since 2014 on biotech workforce education. In part, that's because in the family, I have an educator, my wife, who was teaching a lot of bioinformatics. In fact, we've traded off working in our respective companies over our career. So she was at Geospiza for about 10 years, did a lot of community development, and got into doing courses in bioinformatics, and then started digital world biology. And so when I left PerkinElmer, I had a place to go, if you will. And we work with a national network of community colleges that do biotech workforce through an NSF-funded center, Innovate Bio. And through the course of things, developed a website about careers. And you're going to see some examples of this as we go. But then I also do a lot of consulting. So I work with the University of Washington Tech Transfer, where a couple of companies I now serve as an advisor to phase genomics, kind of classic genomics, except there is no such thing as classic genomics now. So they combine a Hi-C method so that you can combine fragments of DNA together that are in close proximity, and then use a lot of machine learning and these kinds of technologies to then develop and construct genome maps, depending on what the sample input is. And then Wayfinder Biosciences is a new company in synthetic biology who are looking at RNA aptamers and developing molecular sensors so that you can optimize synthetic biology chemistry. And one of their fundamental problems is how do you explore RNA sequence space and come up with molecules that can bind chemicals and then use those as control systems. So if we have time and some Q&A, you can ask about those companies, but that's all I'm going to say right now. So with that, what I want to do is cover my development and some of the things that I think about and I've learned over my career. So this is, with hindsight, is, what's important, right? So, analysis and models, process automation. You're going to see a lot of themes around process automation, but really the quality control. How do you know the input data are good? And how do you use outputs to understand the data? Visualization becomes very important in that. And then software development and user interfaces. I mentioned Finch TV as an example. Databases are very central to a lot of what I do and think about. And then finally, the world in genomic bioinformatics and structural biology. So it really began for me in 85 with Excel 2.0 and a Macintosh, okay? And I was at Genetic Systems, and we were working on LAV ELISAs. So ELISAs are enzyme-linked immunosorbent assays. And LAV was the precursor name or the competing name to HIV at the time. So lymphoadenopathy virus. Luc Montagnier, he just recently passed away. And in fact, he came through Genetic Systems once, so I got to shake his hand. It was pretty cool. And so this was, I guess the one of the last pandemics with HIV. It was a very, it still is. People can manage the disease now, but back then it was killing a lot of people, and so the tests were very important. And what we would do is we would work on developing immunoassays, and my computation was I learned how to use Excel. I learned how to do a bunch of copying and pasting, and I learned how to record that and make macros. And so I literally reduced my job to running my assays in the morning, reading the data into the computer, taking single rows of information, converting them to grids, I knew what experiment I did. And so I'd have just the grids made, and then I'd select the data from the appropriate grid to either get an optimization plot or a histogram or something like that. So we could look at sensitivity, specificity, standard deviations, and develop our assay. And so that was the beginning of my, my career in data processing. I like to say I bored myself out of a job because I got it so routine that, I couldn't stand doing it anymore. So I went to another company called Microprobe. And this one was kind of cool because this gave me a glimpse into molecular modeling. And we were also, as a diagnostic company, we were working on oral pathogens. And so I was, I could bring that assay development experience. But then we also did this, our research chemist director, he was into antisense oligos. And what was sort of interesting about that was the field itself was in the late 80s, everyone was like, antisense, this is going to be the new treatment. And if you look at, this table, it has us going from antisense in 98 to 2013 is the next approval. And in fact, this particular antisense oligo didn't work by antisense. It worked by stimulating the immune system. That's a different story. But being an experimentalist, my job was to design an oligo so that we could take an oligo that had a moiety in it with this iodine group. This is a leaving group in chemistry. So guanine, a lone pair on the nitrogen, could attack this, and you could get a cross-link between two oligos. And I got the chemist really mad when I did this experiment, because what I did is I put 2 Gs in. So I designed the experiment for success. And he said, hey, wait a minute, it's not hitting the right G. And I said, well, but it's hitting this one. We got a cross-link, we can optimize later. And he said, but my model said it should hit here. Well, he went back and re-looked at the model, and there was a hydrogen bond that actually moved this down and hit that, or moved it up to hit this G. So the point is you got to design your experiments, so that you can get a signal. And the other thing is and again, a lot of this prescience, if you will, is with the perspective of hindsight. But there's this famous quote, "All models are wrong, but some are useful," by George Box. And doing some research, okay, did he really say that? You go on Google, and somebody wrote a Medium blog in 2019 that said, this is what he really said was, remember that all models are wrong. The practical question is, how wrong do they have to be to not be useful? That seems like a reasonable thing an academic would say. So this has got to be the right quote, right? But there's another literature reference that you can go and get the book in Google Books. And right there in the heading, it says all models are wrong, but some are useful. So you got to do your research and verify your facts. But that model was a little bit wrong, but it was very useful because it got us on the path to something. And so that was kind of, a quick journey through my 5 years of being a technician. Then I went to graduate school in '89, and I finished in '93, largely because I had this industry experience. And I worked on natural product biosynthesis. So if you think of antibiotics, you think there's anti-cancer compounds, natural products are a really big deal. And at that time, we knew how they were made from a lot of chemistry experiments. You'd feed bacteria different kinds of chemicals, and they'd reassemble them and put them together, and we use NMR to figure out how they got assembled. So when I joined, I had this experience in cloning and stuff, and it was time to get after the genes. So that was kind of my job, was to try to figure out what the genes were that made it. And when I began graduate school, by the time I finished, we went from sequencing the hard way with S35, where you still did, same kinds of bioinformatics we do today, but it took 2 people to read these gels. One would read the gel and the other would type it into the computer. People were working on ways to read these gels, but then we had automated sequencing come out. And this was the invention out of Lee Hood's lab. And that's where I lined up my postdoc. So I got to, in 6 months, get more sequence data than I had ever gotten all of my 3 years in graduate school. And with that, I also learned that the model we had for biosynthesis was the resistance gene would be with the biosynthetic genes. And in this case, the key resistance gene, because a bacteria that makes an antibiotic needs to be resistant to its own compound, the gene was not with those genes. So that model was incorrect as well. But this is where I learned how to do command line. So, done with Excel. Now I'm on the VAX. I'm learning about batch processing, and that was pretty cool. It was so cool. People started to bring me data and said, "Could you analyze this for me? I don't have time." And so I was getting co-authorships and hits that way, and through the process, I had an idea of how this was the antibiotic I was studying. It's a peptide antibiotic. Everyone recognizes that, right, from the structure. And these are the amino acids. And so what we thought was, much like an antibiotic like erythromycin is made, and much like fatty acids are made, there was evidence of this in the literature for other antibiotics, that there'd be a large enzyme system that would move amino acids around and assemble them and move the chain around, and then that would be modified. And so this was a conjecture I had, and it was the subject, I think, of chapter 2 of my thesis, was how this antibiotic was made without any scientific evidence, because the genes were really elusive to try to get. And part of that, and this is just showing what we've learned about polyketide synthetases since then. So, since I finished and now, we have tools like cryo-electron microscopy, so we can really interrogate structures. And so this is just in color showing that you have these different modules. And this is what we thought was going on with the peptides. And I'm saying that a lot because what we learned in 2009, so I'm way past graduate school, 15 years later, we learned that the biosynthesis was really ribosomal. That is, that little peptide is made, and then the enzymes come in and modify it. And we did know that the antibiotic bound to our ribosomal RNA in protein L11 of the 50S ribosome, but we never had a picture like this when I was doing my PhD. And since then, people have, these thiopeptides are compounds people are interested in, and they're so interested in them that by 2012, a group had made a website so you could put in a sequence. In fact, when I first learned that my main hypothesis in my thesis was wrong, was going to a seminar at the University of Washington by this fellow Fischbach, where they were studying gut microbes and finding all of these thiopeptide genes in them, and there's even a company that has developed to allow you to make benchtop biosynthesis of thiopeptides. And so I show this just as an example of something you're going to experience in life. In 20 years, you're going to look back at the things you're doing now and go, wow, they were so simple, so quaint, and so that's it's kind of a neat thing to do. So that was graduate school. When I then went and did my postdoc in Lee Hood's lab, I really moved from the bench to the computer. So I like to tell people I did my last bench experiment. It was February something, now it's becoming a blur, but it was somewhere around 1995, and I had to close a gap in the BRCA1 gene. And there are two things that I did that I think were significant is I sequenced the BRCA1 gene. So that's just something that people think is really cool. And I did that in collaboration with Mary Claire King and Lee Hood. And the other is I started to get into data processing. And, from my bench experience at Genetic Systems and assay development, I got into large scale sequencing. And one of the things I noticed was we weren't very methodical about it. And part of that is we didn't have a way to summarize the data. In fact, this predates Fred, this paper, and Fred was the base calling algorithm that Phil Green developed that I think people often think of FRAP, which is the assembly algorithm, as a really important contribution, and it was. But Fred was the major contribution because what it did is it assigned quality values to DNA bases. And those quality values, get referred to in Illumina sequencing sometimes as Fred quality values. And it was the ability to have a probability associated with a nucleotide in a sequence that said this one is probably correct or not, and have that be on a scale and have that be systematic, right? And so I developed, got onto Unix, started to learn the Unix pipe, started to learn Perl, these kinds of things. and developed a system where we would get data onto the computer. We'd sort it out because we'd put metadata into the files. We could read the files, and we would do a bunch of data processing, and then we could send a report to everybody at the end of sequencing. And actually, that improved sequencing more than any other. Was everybody got the email at the same time that said how we did. And this is an example from the paper that showed that we really had a process out of control. So in statistical process control, if you have a aggregation of data, and this is 900 chromatogram files or 900 reads, and then you look at instances of a plate. So this is 9 plates that went in here. We'd have one instance, 51 of these would pass, another 60. And you can see that these distributions are all over the place. What that tells you is you have things going on in your laboratory process that you can fix. What you want to see is everything have the same shape, because then your variance in the data is governed by stochastic variation, which is a statistically normal thing, right? So that was, that led to what we're going to do at Geospiza. And then BRCA1 was a scientific project that and developed a program that got me a cover of Genome Research. It was also having, I think, Mary Claire King on the team help get that cover of Genome Research, but it's something I'm very proud of because it's kind of cool to get a cover. And what I built was a program called DrawMap. And this was, this predated genome browsers. So you're familiar with genome browsers. This is a program that the output is actually Postscript. So I learned how to program in Postscript. It's a real language. And I had Perl tools that would parse data and create graph types. And then this display, all the logic was encoded in there, including the figure legends. So I had different colors. And the BRCA1 gene is this part in the pink here with a very large exon in the middle. You can see it spans 80 kilobases of DNA, and it's about a 5 kb transcript that gets produced to create a 1500 amino acid protein. We also had a couple other proteins. Genes, they're just here. We don't know why. CPG islands were something we were always curious about at the time. You can see that I have some big ones in front of the genes because CPGs were felt to be, and in many cases are associated with promoters. What I really want to highlight is tracks F and G. These were output from gene finders. So gene finders could kind of find a bit of the exon, or they might split up the exon. You can see down here, they didn't find these very well. Well, what was learned about the early gene finding algorithms is they were trained on data from things that had been cloned. Well, it turns out at that time of the world, we only cloned the things we knew how to clone that were easy to clone. Well, those also were things that you could find genes on. And so the gene finders weren't that good when you got into anonymous DNA. I think they've improved now because we have more data. And so it's again, coming back to that, all models are wrong, some are useful. At that time, they weren't that useful. But one of the things I could do with this, getting to process automation, and this is just, I got to a point where I got to version 3 of DrawMap, and I had really thought about the architecture of it and what we were doing. We're taking data from different programs, having to put it into a common structure so we can feed it to different kinds of graphing tools. And then that has to get combined together to write the lines into a Postscript program. But then also I needed to, deal with how I passed in my parameters. And so I would do this structure, and when I give this presentation at meetings, I would, have people come and ask me for my business cards. So that was very gratifying. So computational biology is a good place to work, I think. But what I could do with that is, at the time, again, there weren't that many sequences that were greater than 50 kb in length. So I went to a database and I found this list, and I got the sequences, and I built a system that would send the data to BLAST. It would pull back the hits. It would create, metadata or data sets of those, run another alignment, and then ultimately send everything to a printer. Okay, so what we did is we used the printer and we made a book, and then we looked through for interesting stories. And I had 2 slides. One is a Titan. And what I'm showing on these axes, these are actually expression hits. And then in the third dimension is the percent identity. So you're getting kind of a picture, instead of a stacked plot, you're getting a little bit more depth. And so titans are proteins that we have in heart tissue, and they're full of these Ig domains, which is actually, we have a colleague at NCI who's really interested in studying them because they also connect neurons and brains and things like that. And they're also in antibodies. You're going to see a thread of antibodies through my career. And then this one here is the human growth hormone, where I could look at these 5 different clusters from express tags, EST data, we called them then, now it'd be like RNA-seq. And it was kind of cool because you could see these gene families really well. What's sort of interesting about this and sort of sad is if you did this today, you couldn't see any of this because there's too much data in the database. The whole reason why this worked is because I was at this part of the exponential curve. So one of the themes here is the data are going to continue to increase in an exponential fashion. You are here, but in 20 years, it's going to look like you are here, right? And so the observations that are made, the insights we get change as we get more information, okay? And because I was working there, my DrawMap experiment worked very well. And I had repeated it, I don't know, back in this time frame, and it was like, wow, it's just all red mush. Couldn't do it anymore. So as I finished my postdoc, I had really kind of moved from, doing those, early days of Excel to a command line to automation systems to now projects that involve putting information into databases and mining that data. So, you've got a lot of blast hits. You want to mine those for things, and working with software engineers. And that's why I put, by the end, it was, I was starting to think about databases, the architecture, and teams to get projects done. So one of my colleagues, he built me this layer right here, dbi.pm and oroperl.pm to communicate with the Oracle database. And that gave me an API where I could then create all of these other programs to take my automation systems and load tables in Oracle. We were fortunate enough to take an Oracle class, and there was this one thing the instructor was talking about, dropping your constraints to load a database fast. And she was saying, you got to make sure your data are clean. And I'm sitting there puzzled. What do you mean by my data are clean? How are data dirty? Well, I quickly learned as I started to load data into that Oracle database, how quickly things would blow up for things you didn't expect to be in the data. You would have assumptions about how long a read would be or how long a sequence would be. You would write a table that would have that column width. And then all of a sudden, it could be the 100th sequence, it could be the 5,000th. It's as you clean your data, the frequency of error starts to decrease, and that's where you start to learn about, the architecture of having a software system that can do rollbacks and things like that. So you don't have fatal kinds of mistakes. And so that's sort of where I ended up as a postdoc. Was I had a few primary publications, but I also had co-authorships. People were bringing me stuff saying, can you help me? Can you help me draw that picture? Can you help me, get this stuff annotated? And it was really, I like to say I had more of a service than a research project. And through a circuitous route, ended up founding Geospiza, which, the idea at the beginning was we're going to help people assemble genomes, but we also had to do the lab tracking. And that led to the development of a fairly extensive software system. And what I'm showing here is the timeline, so Geospiza was from 97 to 2011, and then PerkinElmer acquired us, and I stayed on to 2013. And so that was this period in here. And what I've highlighted up on top is like the first papers in pyrosequencing, microarrays, capillary electrophoresis, DNA colony massively parallel sequencing. This is a really important concept. And this is like 98. And, but you can see that, microarray papers aren't coming out really until much later, and next generation or deep sequencing doesn't come out till like 2008. So the discovery and the invention of a technology precedes the use of that technology by a good 10, maybe 20 years sometime. And then I have the instruments that are introduced. Some of these, Solexa is now Illumina. Illumina bought Solexa. So this is the short read sequencing that we're pretty familiar with. And by 2013, when I was working on a paper with Chris Mason, we noted like 18 different types of instruments and 10 sequencing modes. And so we started with Sanger sequencing. So, all of that stuff I did with Hopper is now behind a web interface. We've been introduced to core laboratories. These are people who need to be able to take an order for sequencing. They need to be able to process the samples, collect the data, have email tracking, review it, and then post that data for their customers, right? And so we built a web system that it turned out was really ideal for that. And you can do things. This is just showing, in the sequencing, various kinds of artifacts that can occur with repeats, showing how you get truncated sequences. And then this heat map is taking a compilation of data and some SQL to assign some values and then displaying it in Excel. But the basic idea was this was my Hopper system that I did as a postdoc. That was all command line and pretty crude. And now it's a full-on software system with a database behind it and a web interface for entering information. But then one of the things happened, as I was noting in that graph, was disruption. And this is I love this plot because when this first started, people were saying sequencing costs now are dropping faster than Moore's law. And I like to point out that sort of, okay, what happened here was we changed the format. The format going from sequencing one sample at a time to being able to do massively parallel sequencing where we collect billions of reads at a time changed and made this discontinuous. But you can see here, once we're on a platform, it kind of follows Moore's law. All right, so if you want to know where this is going, you can just keep drawing this line and then map it out. The other point I like to make on these things too is we say, well, we have this cost per raw megabase of DNA sequence. Well, yeah, it seems really cheap, right? Except you got to buy these things a billion at a time. You can't get like 1 megabase of sequence. I can't get, spend 10 cents or a penny and say, can I have some sequence data? It's like, no, you got to spend $10,000, but you get a lot, right? And fundamentally, what happens now is sequencing becomes an assay, right? So we've gone from this kind of concept to single samples, chromatogram kinds of files, to now massively parallel data sets. And when you do that, you start to change science, right? So I did this blog in 2010. So one of the things I've done through my career is write about these things as I'm experiencing them and then putting my spin on them. But you have, in the early days, we did the cloning, restriction mapping, to genomic sequencing. This is very hard and arduous. The first completion of the human genome was done this way. But then we get massively parallel. And now we can think of how we do de novo, which is, doing more genomes, metagenomes, going to dirt, sequencing dirt and asking what's in dirt, what organisms are there, to then functional genomics, looking at gene expression at higher throughput regulation. And then we start getting some really kind of, cool names assigned to these assays. And then of course, variations. So that's 2010. 2013, wow. People have been busy. Jay Shanduri of the University of Washington has brilliantly analyzed this in the context of a subway map, putting together all the different methods that would go with these. And Hi-C, you can see Hi-C is in here. You can see Immuno-seq. And then I just kind of exploded the diagram. And today we talk about single cells and we talk about spatial cells, right? So can I sequence cells within a tissue and then look at where things are expressed in the context of the space and surroundings that they're in. So that was pretty cool. But to do all these things, you got all these complicated laboratory processes. And I mentioned that you're going to have to buy reads a billion at a time. And so you have a lane of an Illumina flow cell. And so for us, the challenge was, well, I got samples on a plate. I'm going to make libraries, and I might combine a bunch of these into a tube and put them on a lane, or I might want just really serious read depth and take and put single things on a lane. And so we had to develop all that tracking to be able to do that. We also had to be able to put in the laboratory data, right? So I'm doing my library preps. There's things you want to do for RNA, like RIN analysis, and you want to then later correlate this data you collected in the laboratory with the final data. And so to do that, you need a laboratory information management system, and that has to be very flexible in terms of how you construct these various workflows and link things together and allow users to define the rules for their process and have the software be able to enable it. And then somebody says, well, we have a clinical lab. Can you do this for regulatory? So then you got to put in a whole change control system and all of those kinds of things. And so ultimately what we really did at Geospiza was link analysis. We also acquired a technology called Gene Sifter that was very good for microarrays. And we converted that to RNA-seq, because once you've reduced your data to counts, it's gene expression, right? And so we had, this would be like the laboratory data, the quality values that we saw from the early chromats, and then the analysis together, and we could do a lot of these different kinds of assays, and we developed a framework. And when this happened, people would talk about primary, secondary, tertiary, quaternary analysis. In computer science, we'd call it level 0, level 1, level 2 data. Fundamentally, what I learned was nobody knew what we were talking about. We were just talking to ourselves, right? So we got to talk to people. So the first thing is you're collecting the data, right? So you'd have these images. I'm not going through how the massively parallel sequencing works, but basically it would be these images you would read successively. Something does the image analysis and you get bases, right? And these bases have quality values. And then what you need to do is reduce that, right? Reduce it to something meaningful that represents the experiment that you were doing. So a lot of times for next generation sequencing, it's reducing the data to counts. We align our reads to regions of a genome, and then we say, okay, how many did you have in that spot? And then there's nuance on top of that, but that's reducing the data. And then you have to compare between many samples. This is how you start to get to insights. Do you see in one set of samples and controls, samples higher gene expression than in the controls, and can we use PCA plots and things like that to decipher this? And then you need to take this insight and apply it to a context, like a pathway map of genes, so that you can get to various kinds of ontologies and pathways and networks, and ultimately develop new knowledge, right? So for here you get your observation of how your samples differ, but this is how you get the meaning of that. And so really this is a process that hasn't changed, right? What's changed is the instruments change, some of the experiments change, some of the questions we might ask change, but the overall process actually stays relatively the same. And so that's somewhat gratifying. And so that's kind of where that ended. One of the things along the way, we needed to develop, we were very early in next gen because we had the sequencing experience. We could really move to that quickly. And I wrote a grant, an STTR grant, which is a way businesses can get money from NIH, SBIR and STTR, small business innovation research or small business technology transfer, to take HDF5. HDF5 is hierarchical data format that came out of really physics and engineering. And the things it does really well, it's very fast. Its speed of IO is at the level of disk writes on your computer. And it handles sparse data matrices. One of the things sometimes you learn when you're doing databases is the size of your data is mostly empty space. What if I could have a table that all of my zeros could be compressed and I'm only looking at real data values? And so HDF solved a lot of those problems, and it got us very quick. We got to solutions very quick that we're using a web-based interface, and it's snappy. I can go from alignments and a picture all the way down to the individual reads with a few clicks few clicks without any real significant latency latency. So it was really huge. But one of the things that happened was, we lost to BAM, the BAM format, that came out of Broad, because Geospiza wasn't Broad. And, and so, as if I summarize that in this first part, what were some of the lessons I've learned in this? One is and you'll hear people when you talk about bioinformatics business saying they're really fragmented and competitive. Well, technology changes all the time. Algorithms are critical, but I'll say that many have short half-lives, except for BLAST. BLAST has persisted beyond anybody's imagination. And data integration is often harder than it sounds. And finally, for a tech company, its inventory is innovation. And I'm going to zip through some of this pretty fast, but one of the things I want to make is a point that technology, information advance is exponential. So, we're seeing the explosion of methods, data, even databases, although this is kind of artificial, this plateauing, because these are databases that are in the Nucleic Acids Research Archive, and not all databases in the world. But this is really, these are knowledge resources. The market fragmentation, this is Biotech Careers, which is a database that has a lot of biotech employers in it. And as of in 2011, we had about 100 bioinformatics competitors. Now there's about 318 that we've noted in our database. And so today, what I do is a variety of consulting. And these have been projects that have involved other people's databases, synthetic biology. One cool one I did some work on a Deep Tracer, which is an AI algorithm that is taking and using AI approaches to take cryo-electron microscope maps to models with very, very, very refined models very quickly. And then Phase and Wayfinder I mentioned. And so I also advise on, a variety of things and help with grants. And so one of the things, that I've noticed over time is, one is the data abundance is driving a lot of machine learning and AI. Architectures themselves are encapsulating their complexity in containers. But many of the old problems persist, especially with respect to data automation and data use. And in fact, NCI, NAID, and other institutes at NIH fund major collection projects, but the resulting data resources are not widely used. And then they come back and they do requests for proposals for, say, SBIR contracts for companies to make use of these data. And one of the things we still can't do is add human insights to data. And education has been really slow in adopting and teaching computation in biology. And so with that, digital world biology, we're trying to do something about that. And so we have there's general bioinformatics that Sandy's been teaching for a long time. And then I'm just going to give a little bit of a glimpse of something we were doing with Shoreline Community College on immuno-bioinformatics. And we also have this very popular BLAST tutorial because of what I said earlier. So this was a presentation I gave in 2019. Now I'm just going to touch on a few of the highlights. First, let you know that we're funded a lot with NSF and Advanced Technological Education. So NSF supports the education in high-tech fields that drive our nation's economy. So biotech is one of them. We're always working on bringing bioinformatics into that. It's a lot of lab work. And so immuno-bioinformatics is also called immunoprofiling. And this is the idea that in a drop of blood, we can interrogate an individual's immune system. And so it's being used extensively in diagnostics and biomarker discovery. And the idea here was to use a refined resource to teach some of the principles of immunoprofiling, and then use tools like CyVerse and command line to teach the ideas of the underlying technology that go into that. And so immunoprofiling, this is a blog I did, and I love it because if you do this search on Google, it comes up very high, and it has publications per year, but it's the idea that you can drop of blood, sample all the immune systems, immune cells, and therefore the antibodies or T cells. And why is that important? Our antibodies are, we have a tremendous abundance or repertoire of antibodies that can be produced because of the gene segments and how they rearrange to form complete genes, and then at the mRNA level. So in your B cells or T cells, this process is going on, and you're producing these molecules. And when you are stimulated with an antigen, like the COVID virus, you're going to start to produce antibodies, and then they're going to start to refine through a different process. And so if you have like a single drop of blood, you can find these are a special kind of T cell, but there's going to be 10 million red blood cells, but 100,000 white blood cells. So these are the ones producing antibodies, and 5,000 conventional T cells. Well, what if you have 2 drops of blood, right? So you could sample that repertoire with high throughput sequencing. And the way it works is you collect the sample, collect the blood, and then you have an assay that samples the region of highest diversity. So using PCR and sequencing, you can get into this small area here, and then you can start to look at things, right? So in this particular class, what we were talking about doing is taking a paper that came out of Steve Quake's lab, and there's 3 million reads from 29 or 49 different samples. So it was really 3 samplings from an individual, blood, normal breast tissue that's cancerous or precancerous, and then looking at the T cells. The 49th was one of those was done twice for a technical replicate. And basically there were 10 assertions in the abstract. And so using their system that's already done the alignment part and put the sequences together, can you use their software to figure out how they got to these particular answers in their paper? And developing this, I had to do that myself, and it was a bit of work. So it's how science is done, right? And what they learned was tumors have a higher clonality, and so you have to figure out which analysis method to use to get to these kinds of things. Another was 50% of the TCR sequences are shared between tumor and normal in the same patient, and so they have some tools that allow you to lay out the different plots so that you can see the different pairs. So each unit of 2 is a tumor normal pair. And so if you go down the diagonal and color code by the percent match, so blue is very low percent, what you then see is that the TCR sequences aren't shared between individuals, they're within an individual. And there's differences between a tumor and normal, but that it's their T cells. You have your own T cells, right? And then these are just the cluster plots that present this data. And then, through the software, you can get into these details. And what I didn't tell you about the adaptive system is what we're using is a publicly available interface. So part of their business, they do this for people is they do the immune profiling, but they have a massive database with all kinds of publicly available sets. And so you can use this in however you want, and so that was the idea for this class. And what's kind of cool too is you can take these sequences and you can say, some of these is when I gave this talk, I said, Google is the new blast, because you can take a sequence and put it in Google and then find out if it's been something that's been discovered in somebody else's, there's in the patent database. I'm not going to talk about the immune repertoire database, but that was another piece we integrated into this. And then the core bioinformatics was in CyVerse. And the idea here was to give people, give students a flavor for how to do Unix, difference between FASTQ, FASTA. IgBlast is an underlying algorithm that's used to do the immune alignments because you need to take into account the V, D, and J segments as you put things together. And so you could, with CyVerse, you could have all these different instances. A student could have their own computer, and every time you do one of these things, you go out and start to put it together, and it's a new adventure. So I wrote a blog that was my documentation for this, but also expressed how I felt about going to sea again and finding all these tools and compiling all the data that we needed to put together. So so you can read that at your leisure. And then here's the real killer, right? So this is a command line. Who can ever type that right the first time? So just that alone is instructive. But then after you do it, what you're going to get is you're going to get output, but this output is tab separated. So you can take that output and you can put it into something like a Google Sheet. And then you can take that Google Sheet and click the Explore tools. And all of a sudden you see that you're starting to get, all the same things that we saw in the adaptive system. So it's really showing how it works under the hood, but giving students experience into, touching the bits, if you will. And so you can even get, to a count of the different gene families or genes that are assembled together and their frequency. And there's a lot of benefits for the students. It's immunology is it's about 20% of biotech. I might have that on a slide. And, so for students that are coming into a college setting and are going to go on to industry, having this kind of experience is really, really important. This has led to another grant that we have on antibody engineering, where we're exploring hackathons to develop course-based undergraduate research experiences using bioinformatics, IC&3D, and IEDB in education. And the reason we're using IC&3D, I know Taru talked about, we have this iPad app that we have a structure viewer on, and it's really beautiful and great to work with, but this is web accessible, and it connects really all the different layers of information, the DNA sequence, protein sequence, to the secondary structure, to the tertiary, to the quaternary, in a really great way. And this is in the center here, we're looking at, and you probably can't see my mouse, but I didn't turn the pointer on, sorry about that. The epitope-paratope interface. So this is where the antibody binds the antigen. And then you can look at the interactions, and then you can change the amino acids, and there's mutation prediction algorithms in the program, and so you can see how that can affect the binding. And in fact, we're doing a project from a hackathon we participated last summer to look at how mutations could impact these interactions in SARS-CoV-2. You really can't do anything these days without talking about SARS-CoV-2, right? The other thing about IC&3D is it's completely scriptable. So you can annotate structures, color them, and do different things with text files. And you can do this in a lot of the different programs, PyMol, BDM, and so forth. But this one's 100% web-based, so it makes it very accessible for the class. And then, just about at the end here, we might have a couple minutes, but we always think students need to learn their fundamentals. So we've developed some card games to learn your amino acids and learn properties about amino acids. And then they need to know about jobs. And so this is the Biotech Careers Database that has a lot of information about what it's like to work in biotech, but it has this employer database that we maintain and allows us to look at data in different ways. So like the picture I showed you of bioinformatics and genomics, we can count, because we have keywords for the employers, and we have like close to 500 keywords that describe the industry and what people work on. And so that's kind of cool. So, in summary, so how has computing changed for me anyway? 97, these were the tools. 2022, I don't know if anybody's using Julia, but I read about it. We argued about PHP. The Biotech career site is on Drupal. If you look at the internet, WordPress, Drupal, Joomla, several other content management system platforms that people build websites with are all PHP based. In fact, in bioinformatics, there's the triple community that do model organism databases on top of Drupal, and so they have to learn a lot of PHP. But JavaScript for doing a lot of interactive kinds of things on the web. And the other is that Google, I don't program every day, but I know the questions I need to ask. And boy, you can sure, find ways to solve problems by doing searches. And so where's this going, right? So, 3D molecules, that I was showing in IC&3D, people are looking at how to do these on whole systems with molecular tomography. People are designing biomolecules, antibodies from first principles. So can we just order a sequence and get a functional protein? That is happening. So there's a lot of things that are going to be changing What do the next 20 years look like are going to be themes on this, including many more V1 systems. And this was a seminar I attended, a workshop back in November. And I really liked how they broke down these life phases in the data life cycle, and then talked about the different roles people have in terms of what they do, the questions that are asked, who poses the question, who deals with the question, what are the pieces that go into that. And so I redrew that and did a blog on that on Biotech Careers. And so with that, I want to thank you. I know I was running a little late there.
No, it was great, Todd. Thank you so much.